I got my PhD in 1995. I've worked on a wide range of plant viral diseases, and I've met folks. I joined Texas A&M about 5 years ago, and I've been working on a wide range of problems, and I got involved with Latent Viroid when I met folks from Rare Earth Genomics and started working with Russ Jessup, who's a breeder at Texas A&M, to try to tackle some of these questions and issues. And while I could say I'm an expert at plant virology, I'm also feeling like I'm an expert at something in Latent Viroid we know very little about. So I'm excited to be here, and I'm excited to take on this project. There we go. So we've developed an infectious clone of Latent Viroid, and this is actually my starting talk. I am in the plant pathology department at Texas A&M, and this is an example of some plants that we infected in our growth chamber. And one of the biggest questions that we have is you can look at these plants and the same variety has different disease patterns. So how do we explain the disease? How do we conquer the disease? How do we recognize the disease? There's one healthy plant here in the corner, and you can see very severely chlorotic plants and mildly chlorotic plants, and they were all inoculated at the same time. And so this creates a challenge in itself for working with growers, trying to help them identify healthy from infected materials. And I wanted to start by telling you a little bit more about the life cycle of viroids. So it's very easy for non-virologists to think a viroid and a virus are the same thing. They have the same life cycle, and they really are quite different. Viroids are small circular RNA molecules that don't encode for any protein. They're only 250 nucleotides in length, which is crazy small. I work on RNA viruses that are 10,000 base pairs in length. So 250 is about as small of an infectious agent as you can get. Because they don't encode any proteins, they are kind of a miracle, I think, because how do they cause such an amazing disease and cause so much changes to a cell and to a plant without really encoding anything at all? Latent Viroid is a circular RNA. This is its replication cycle. It encodes a single-strand nucleic acid, and it replicates in the plant cell nucleus. So it depends upon the host machinery in order to replicate and cause infection. And so the host RNA polymerase acts on the genome and produces a copy, and that copy then serves as a template for production of multiple strands. And each of those are produced and circularized, and it continues through a cycle of synthesis and replication. And it's able to spread throughout the plant, so that means it must exit the nucleus, move through plasmodesmata between cells, enter the vascular system, and somehow wreak havoc on these plants. Latent Viroid causes dud disease, in case you don't know. And so it's interesting for me as a plant virologist to try to understand how a 250 nucleotide amino acid can cause a latent infection where you don't see any symptoms for a while and then suddenly destroy the flowering ability and affect its complete development. So viroids, many viroids, not just Latent Viroid, are latent viruses. They are latent diseases. It's quite common amongst the viroid pathogens that they cause latent infections, and then you don't see them until much later. One example is peach latent viroid. There's huge problems of viroids in stone fruits. Peach is one example, and you don't see disease until the trees start to wither. So because of their issues in stone fruits as well as in hops and in hemp, they are an economically very important category of pathogens. And as you know, Latent Viroid causes extremely important, significant problems in the hemp industry, which is how we got involved in this. And so as we got involved about a year and a half, 2 years ago, working on hop latent viroid, we started developing the tools to detect it. One of our first most important questions was early detection of hop latent viroid in infected plants. And how do you know if you, and the main approach is to use a PCR technology. And we're very concerned about quality control in our research and in our efforts and making sure that we get true positive results, not false positive results. True negative results, not false negative results. So when you're dealing with a plant with such an array of no symptoms to severe chlorosis and death, how do you know that you might have missed a positive outcome? And so one of our PCR technologies has allowed us to identify the genome, which is 256 nucleotides, as well as replicating intermediates, which can be 500 to 750 up to 1,000. This viroid, as it replicates, produces multimers of itself, and then they cleave into mature products. So we've been able to detect the replicating units, both as monomers, dimers, and trimers. And through this technology, we've also developed infectious clones that are themselves monomers, dimers, and trimers, and are using that to inoculate plants so we can begin to study uniformity of inoculation, uniformity of infection, and start to provide real workflows for how to identify the problem, how to solve the problems, how to make sure your mother plants are clean. So our major goal right now is to understand, some of the things we're interested in is understanding non-host resistance as a guidance for design and viroid-resistant plants. We're looking at screening germplasms to identify germplasms that may not be susceptible to hop latent viroid, that would be non-host, and compare them and perform crosses with very susceptible plants and see if we can understand the genetic basis of resistance. We're wanting to understand what happens in the host during latency that's different from what happens to the host during severe symptoms. What is the viroid doing? Is it just dormant, not doing anything, or is it doing something metabolically that the host actually is having an immune response? Maybe after latency, that immune response is failing. So we need to understand what is the host response and how immunity functions against viroid diseases. And then we'd like to engineer viroid resistance using a range of novel technologies. And so these questions that we're asking, they're also asking in a wide range of other viroid diseases globally. The problems that we're facing in hemp with hop latent viroid are the same problems they're facing in stone fruits like apricot, plums, peaches. And so what I also wanted to start my talk was differentiate hop latent viroid from hop stunt viroid. I found in talking with folks that it's very easy to mix the two up. They are actually two different species of viroids. They both infect hops. They both seem to cause a stunting when they cause disease. But hop stunt viroid is a different organism together. Hop stunt viroid infects hops, it infects grapevines, it infects citrus, cucumbers, and a range of stone fruits. Hop latent viroid, which is a huge problem for the hemp industry and the cannabis industry, does not infect those hosts. We did a sample when we began this work of over 111 plants, and we found that 80% of those were infected with hop latent viroid, and we found no evidence of hop stunt viroid co-infecting these plants. So we're clearly looking at a single infection problem, not a potential for mixture of the two. And so I wanted to throw in some pictures of folks that are working with us, and again, pictures of our friends at Rare Earth Genomics that are helping to support this research. This is one of the scientists in my program who is speaking with our collaborators, and these are some of the experiments that he's been running. We've been interested in looking at developing sensitive detection methods. We want to perform assessments of viroid-host interactions. We're looking right now at different hop latent viroid variants and comparing the different variants across different plant genotypes and trying to track the progression of the disease. Major fundamental question, are there different strains of hop latent viroid? Is there a severe strain and a mild strain? So we're trying to tease all those questions apart, and it also requires understanding the different genotypes of the hosts that we're working with. And so here's an example of some of our experiments. Perhaps there's different variants between the two different hosts. And so how we're going about solving that is using an infectious clone technology. When we take sap from an infected plant that we get from a grower, we don't know if that infected material that we get from him has a mixture of variants or is a single variant or a single strain. So using our infectious clone technology, we can make one single strain, one single variant, and introduce that into a variety of different genotypes and start to characterize the nature of the symptoms, the nature of the disease. And then we can start adding new layers of different variants and making comparisons between them. So this is some fundamental work that we're performing now. And again, we've identified, we are examining and working with 71 different isolates. We found 41 different mutations across the populations, and we are now looking at how these different mutations influence symptoms. And so this is one of our exciting new PCR diagnostics that we developed. We've developed some PCR primers that overlap a highly conserved region of the viral genome, and it's extremely sensitive. We can take an asymptomatic leaf that we know we infected the plant. We know it's infected, but it's not producing any symptoms. And we've been able to develop a diagnostic PCR that can detect 10 to the minus ninth molecules of infection. And so we're moving forward with our diagnostic analysis. We've compared using infectious sap, and we've also used our infectious clone that we've generated in our laboratory. And we wanted to understand if you use sap from an infected plant that we've taken from the field, is our infectious clone technology as sensitive, as robust of an inoculum as taking sap from an infected plant? And as you can see, when we're looking at inoculated plants at 26 days, 39 days, 60 days, and then when we use our own in-house infection system at 21 and at 42 days, we see 100% of our plants are infected systemically and 100% of our plants are infected locally, whereas the infectious sap is a little slower at producing infection in some hosts. So we're comfortable with our technologies. We're able to move forward. This is a study that we performed using different hosts. We want to understand how many different plant species are infected with latent viroid that are not reported in the literature. In fact, very few people are working on this problem at all. I've traveled through Europe. Everyone I know in Europe is working on hop stunt viroid, which is a huge problem in the hops industry there, and I have yet to meet an academic that's working on latent viroid full-time as their problem. Apparently, Europe is restricting the ability to do this type of work, as well as many states in the U.S. So we're really happy that in Texas we're able to do our studies on latent viroid. We found it infects cucumber, it infects tomato, and it causes some pretty severe death of chrysanthemum. So if you are a vegetable gardener as well as a grower, you may want to be aware of your vegetables as being a reservoir, or your chrysanthemum plants in your flower garden as being a reservoir for disease. This is an example of our PCR diagnostics, so you can see what kind of results we get when we look at our different plant hosts. We get very clean, very robust PCR products across our plants. Those that are negative is our healthy here, just to show you how our data looks. We found in our studies that Arabidopsis plants, which is a model genetic system, Arabidopsis was one of the first major plant genomes to be sequenced, and it is a model like Drosophila is a model for human genetics, mammalian genetics, C. elegans. Arabidopsis is our plant model genetic system that's used globally for dissecting genetics. And we found it's highly susceptible to latent viroid, which is exciting because it's the only viroid that is known to infect Arabidopsis. And it also infects cucumber, and we found that those plants die at maturity. And so this is an example of a Arabidopsis plants that we have growing in pots, and the infected, hop latent viroid infected plants, once they reach maturity, it's called a decline or a dieback disease. The plants don't die until they reach maturity, and then you see the necrosis, the death, the browning tissue just going backwards down towards the base of the plant. For me, that's exciting. It's a novel symptom, and it's a way we can characterize and start using Arabidopsis genetics to study decline. And here's an example of younger plants that we infected with hop latent viroid using different versions of the infectious clone. And with a higher level of inoculum, we can see earlier decline of the tissue. This is a healthy plant, and then all of these declined quite rapidly before they could produce flowers. So again, here's our mock, and here's our a mutant version of hop latent viroid that's not supposed to produce a dieback, and as you can see, the mutation eliminated the dieback phenotype. So we're excited to now have good diagnostic tools, a good infectious clone technology, and a good genetics that we can begin to look for host, non-host resistance and understand host immunity responses. As we've moved forward, one of the major questions that we wanted to start looking at is developing a workflow for how to sample, how to perform diagnostics in hemp. When you have a latent viroid, where do you sample? If you get a negative result, did you sample the right tissue? Or do you have to keep sampling? How often do you have to keep sampling to make sure that your mother plants are healthy and remain healthy? And so our approach has been to develop a strict inoculation technique where, does that go back? Yes. We inoculate a specific leaf on the plant, and then we start sampling all the way up the plant to identify where does the virus move first. If you inoculate one leaf, 3 weeks later, where is the viroid? 4 weeks later, where is the viroid? 6 weeks later, where is the viroid? And can we track its movement up the plant? Is it doing something unexpected? As you know, as you may know, the literature has shown that this viroid tends to move to the roots first and then moves further up the plant next. And so the best sampling results can often occur by testing roots early and then testing the tops of the plants later. With our new sensitive PCR technology, we've been working at verifying that pathway for virus infection in plants and continuing our PCR testing to streamline a protocol in a way that we're not damaging the plants, but we can reduce the amount of times you have to sample and test. At the same time, we're trying to understand the kinds of symptoms we can see with different variants of the viroids, so we can be the experts at telling you how to manage disease in your crops. So here's an example based on our new knowledge of the dieback symptoms that we see with hop latent viroid in Arabidopsis. We wanted to ask, can we see similar phenotypes happening in hemp? Hemp is what we're working on in Texas. So what we were excited recently, this here is an example of brandy wine variety, or cherry wine variety, I apologize. And you can see that the leaves are starting to die off on the plant, and it's looking pretty sick at maturity. But how do you know if your plant's just senescing naturally, or maybe there's a nutritional issue and it's dieback or not? We're starting to feel more and more comfortable that when we inoculate a plant, we switched over to using another variety called Puma 3. In this variety, you see extreme yellowing of leaves within 2 weeks. We can inoculate a plant, 2 weeks later, we see this extreme yellowing here. And then this is what the top of the plant looks like in other examples, where you get this crazy mosaic pattern on the plants. And so by controlled inoculations and testing different varieties, we can then start reporting what you should see when certain varieties are infected with certain variants of the viroid. So it's exciting for us that with Puma 3, we get this yellowing, and this plant progresses to yellow every week until it just completely turns dead, completely dies. And this is work that's being done by another student in our lab, Sydney Abbott, who's been managing our propagation. She's quite excited to see, this is an example of Puma 3. It's just continuing to yellow. This is the uninfected, healthy version of the plant. You can see the severe differences between infected and healthy. And it's the only time I've seen a grower who's excited that she's gotten an infection. And at first she was scared. She's like, oh, my plants don't look good. I'm like, no, this looks great. This is fantastic. This is what we want to see. So we're proud of the direction that this work is going in. So what we can say right now is we believe that we have, that hop latent viroid causes a dieback disease, that the yellowing and the phenotypes that we see is an example of the symptoms that you can see early. We're starting to work at developing technologies that maybe can pick up on the viroid before you see that extreme dieback, and maybe ways of managing the plants. We're comfortable with saying this dieback disease is what the disease should look like based on studies, for instance, with peach latent mosaic viroid. This is an example of infected peach latent where the plants look great until they don't. You can see the plants look like they don't have very much leaves on them. This is an example of infected peaches versus healthy peaches. I mean, how do you know that you have infected peaches until you started your harvest? Very similar problems to what we're having in the hemp industry. So the viroid problems that we're having here in the hemp industry aren't unique viroid problems to just hemp, but these are global issues that we, as a community within the viroid community, can start discussing amongst each other, and I can pull polls from some of my colleagues working on other crops and work towards common solutions and build on their knowledge to help find solutions for this industry. So we're really excited to be moving in this direction. You can see the differences in the healthy and infected stone fruits, healthy and infected leaves, flowers change colors. All these are symptoms, but they're not obvious symptoms to a grower. They might be anything else. So as we're moving forward, this is an example that I wanted to pull also from peach latent viroid to show you how you've got this 250 nucleotide size pathogen and a single variant which means a change in a couple of nucleotides can completely change the symptoms that you see. So in this example, this is a diagram, this is a diagram of what the peach latent viroid looks like when it's folded into its mature state. And what this shows here in panel B is these 4 nucleotides are changed here. And look at the difference in the symptoms. This is an asymptomatic disease, and this is a complete chlorotic disease. So based on that knowledge, we started looking at a number of informative mutations. And as I've mentioned, we have a total of 41 mutations in our current population of hop latent viroid. We have been performing some bioinformatic analysis to understand which are probably the most informative and useful mutations to go after, to study. And we are starting now to look at the effects of different mutations on dieback and chlorosis in plants. So hopefully this time next year we'll have some updated information for you on what types of symptoms that we see. So I wanted to end my talk the same way that we began, with a thank you to Rare Earth Genomics for their contributions, support, funding for this research. Without them, we would not be even beginning to explore this very important problem. And again, to our colleagues at Texas A&M who have been working in the breeding programs and development of improved genetics, along with Rare Earth Genomics and Texas A&M University. So thank you very much. Any questions? Yes.
How is the viroid transmitted in nature? Transmission of the viroid in nature?
This viroid, we don't even know if there's an insect vector, what the insect vector is. There's really very little work done on this problem, which is shocking. And also for me, quite exciting. I can make some headway on this. We do know that it's extremely infectious. you get one infected plant, it spreads through everything. And to me, that's not much different from tobacco mosaic virus, where the dew or the humidity in a greenhouse can be enough to spread it through the greenhouse, or you get it on your hands and you can transmit it. I would advise, if you have an infected plant, you be wearing gloves and change gloves between individual plants, and take your cutting tools and your pruning tools, bleach them in between plants so that you don't transmit them. We recommend different viricides for treating bench tops and the like to keep everything under control. Yes?
On that picture on the left, and at least one other one that you showed, the control plant that was not inoculated was the smallest one. Is that by chance, or have these all been inoculated at the same time? Or are the different variants? Because it's, you know, what we see in our gardens is that the infected ones are the smallest ones.
Okay, yeah, so good question. These were not clonally propagated. These were grown from seed, and so there's some heterogeneity in the seeds. So.
Investigated some mutants in Arabidopsis, such as replication factors or small RNA molecules transportation within the plants? Have you already used these mutants to address either the replication mechanism or the transport of the molecules within the plants?
So that's some ongoing work that we have going on right now, and I'm not at liberty to speak about it, so yes.
So whenever you're working in a room with infected plants and you're saying change gloves and clean your worktop and bench tops and all of the tools that you're using, I've heard because this viroid is so small that it's very, very hard to kill because of that reason. What is like a protocol that you'll use in your laboratory to stay as sterile as possible?
Very good question. We've just gone through a major clean out. So you see the plants are on the benches there. Between experiments, we bleach the entire bench with 10% bleach. And everyone has to wear a lab coat when they're near our plants. The lab coat stays in the room. You don't leave the room with the lab coat. Gloves stay in the room. All of our tools and shears stay in the room. They don't leave that room, so we're not spreading it from room to room with us. We disinfect our tools regularly. It's usually about 10% bleach, works very well. One of my favorite is cleaners is called Virkon. I use that on the floors quite a bit. They use that also in dairies for making sure that animal facilities are clean as well. It's a commonly used viricide mixed with other cleaners, and we've used it in many greenhouses over the last 2 decades quite successfully. All pots have to be bleached before they're reused or treated with Virkon. And then some of the new cleaners that have come out since COVID work quite well against other viroids. So you can even go to Lowe's and pick up some of the household cleaners that are supposed to be virucidal. I think Jeep is one of the ones that they talk about, and that works also as well, and it's safe for humans. Yes.
So I'm not a botanist or anything, but how closely similar is hops to hemp? Are they like in the same family?
Yes.
Of plants?
They're in the same order. So taxonomically, kingdom, phylum, class, order, family. So they're at the same order, yes.
Just to follow up on the cleanup, when you use your 10% bleach on your tools, about how long do you bleach for, and have you validated any of the timing with your PCR assay?
Let's see. So far we've not had any issues, and it may just be that I've been a virologist for so long that our materials stay pretty clean just from experience. We tend to bleach everything. We have a number of projects in the laboratory as well, so we really are concerned about contamination between projects and between different pathogens. So we do a 20-minute bleach, which is a pretty standard approach, and then everything is autoclaved for us. So all of our tools are autoclaved or autoclavable. So yeah, and since we're running PCRs every day, we run enough controls that we can pick up on things. And then we also, if we feel like it, we will use 0.1 normal sodium hydroxide on our lab benches, which destroys any nucleic acid. And we may do, it, depending upon our situation, we'll do that in our centrifuges as well. I don't like to use bleach in the centrifuges or some of the lab equipment, but put 0.1 normal or 0.01 normal sodium hydroxide kills everything without killing you or damaging the machines. So, okay. Thank you.